Yeah, thanks everyone for coming in. Thanks, Carolyn. So it's my great pleasure to introduce for our this Darwin Day event. And just a quick thing, we've been doing Darwin Day at DePaul since, I saw Jolene there, she can correct me, is it 2012?
Yeah, this is our 10th year.
10th,
This is our 10th Darwin Day.
10th year of Darwin Day. So so it's a really special seminar for us, and I'm glad to see that you were able to join us. And so it's my great pleasure to introduce Dr. Davalos. I met her back in 2008 when she joined the faculty of Stony Brook, where I was a PhD student. So Dr. Davalos got her undergraduate degree from the Universidad del Valle in Colombia, and that is in Cali. Is that correct? Cali, Colombia. And her degree was in biology with an emphasis on genetics. From there, she went to Columbia University in New York. And between 1998 and 2001, she obtained her Master's of Art degree and a Master of Philosophy degree in Ecology and Evolutionary Biology. She also obtained a Certificate of Environmental Policy Studies during that period, so it was a very productive period. Then Dr. Davalos in 2004 got her PhD degree from in ecology, evolution, and environmental biology at Columbia University. So she stayed on there. In 2007, she became a research associate in the Division of Vertebrate Zoology at the American Museum of Natural History, joined the faculty of the Department of Ecology and Evolution at Stony Brook University in New York as an assistant professor in 2008. And in 2015 and 16, she became a consultant for the United Nations Drug Report, their thematic chapter on environmental sustainability. And currently, she's still at Stony Brook University, where she's a full professor now. Incredibly well published, very well funded lab, very exciting research. I always am thrilled to see all the posts from her students and researchers and herself. So with that, we're very excited to have you speak to us.
Oh, thank you, Winsor. Thank you for this lovely introduction, and thank you for inviting me for Darwin Day, which is a growing tradition, I guess, across biology departments, precisely because so little in biology makes sense outside of evolution. We need to understand evolution in order to go forward. So I'm going to be talking about this genomic journey among bats, and I'm thanking, of course, the National Science Foundation and the Department of Ecology and Evolution at Stony Brook University. This is this bat that you're seeing there. It's a little logo that we created for this project, which is a project about sensory evolution. Especially, my lab was especially focused on the genomics of sensory evolution. As Winsor outlined, my lab actually has this double mission on these two ends. On the one hand, I'm interested in how evolutionary processes tend to increase biological diversity. So the center of everything we do has to do with biological diversity. On the other hand, I conduct research about how the process of extinction unfolds, especially in historical times, and that's how I wound up as a consultant for the United Nations, because they were interested to find out how certain social processes dovetail with deforestation or drive deforestation. So in my research, we definitely have this sort of very three interconnected but not identical areas of study. The first one is kind of like the early one since the time of my dissertation. How does biodiversity change in space and time? And then in red, just having to do with extinction, what are the social factors of environmental degradation? How are we in this coupled natural and human system? How does this work? And then in the center, we have biodiversity. What are these biological processes that fuel diversity? And today's talk is going to focus on only one of those three gears. So I'm going to only be focused on the biological processes that fuel biodiversity because it's a Darwin Day talk. And it's about the process of evolution. So since it's a Darwin Day talk, I figured that there was a compulsory Darwin Day quote from Charles Darwin himself from The Origin of Species, right? And we see like this picture of Darwin that I chose mostly because he was relatively young. He was only in his early 30s and with his son, it says, "Can it then be thought improbable, seeing that variations useful to man have undoubtedly occurred, that other variations useful in some way to each being in the great and complex battle of life should sometimes occur in the course of thousands of generations. These preservation of favorable variations and the rejection of injurious variations, I call natural selection. And this is going to be the running theme, this preservation of favorable variations and the rejection of injurious variations, and how we can probe that now in the 21st century, now more than 150 years after we have better tools and a much better understanding that Darwin ever had. Then the second thing that I want to say about that is that, of course, there's Darwin, and then we're celebrating Darwin Day, but of course, Alfred Russel Wallace was a co-discoverer of the theory of evolution by natural selection. And I think in specialized biology audiences, he doesn't quite get overlooked, but I think it's important to recognize that there was more than one person coming up with the same idea and who went on to make a lot of natural history discoveries in his own right. And in fact, for this presentation, I think that Alfred Russel Wallace is more likely to have come across some of the species that I'm going to be showing, some of the bat species, mostly because he worked for wild collecting in the neotropics extensively among the living fauna of birds to sell to support himself, and he lost it all in a shipwreck. So he's also got an incredibly interesting life that somebody needs to make a film about, and it hasn't happened yet. So let's not forget Wallace. Darwin Day important, Wallace also important. And before I get started, I want to thank my collaborators in this great enterprise of this rather large project to try to understand the sensory biology of bats and the evolution of that. Stephen Rossiter from Queen Mary University of London, Karen Sears, now at UCLA, and Betsy DeMone at University of California, Merced, without whom none of this research would have been possible because they were part of the project from the beginning. So let's jump right in, looking at the bats and thinking about the bats, because that's really what drives, like, this passion for diversity is what's driving this. Okay, so when we think about bats, a lot of us that aren't familiar, they're nocturnal animals. They're actually hard to observe. Most of the public is rather unfamiliar with the great diversity of bats that exist. So I decided to put up this slide that shows just, you can see, like, there's diversity in the shape of the ears, there's diversity in the size, there's diversity in the face, the size of the eyes. So there's this huge amount of variation across the radiation of bats that not all of us might be aware of. And what's fascinating about this is that the group of bats that I'm going to be focusing on today, which is highlighted here with the chalk, this group of bats, of all the giant variation that we're seeing in this slide, encompasses fishing bats, bats that are actually vampires, right? Like the only vertebrates that feed exclusively on the blood are included in this group. A bunch of bats that eat nectar, so they're like the bat equivalent of a hummingbird. Bats that are just eating insects, just like many other species of bats. So a single group of bats that's descended from a common ancestor not that long ago encompasses all of this diversity that's highlighted here. We started working on this group a bunch of years ago. So now I guess that would put us, almost 10 years ago. We started trying to understand what were the relationships among all of these very diverse bats that encompass the shape of the skull, the shape of the cranium, encompasses the entire shape variation of all vertebrates in terms of elongation and compression of the skull. So we're trying to figure out, were there drivers of variation? Was there something special in this huge variation? And that took us in the direction of the sensory system from this early work that was just examining the patterns of diversity, how much diversity accumulates over time, finding that there was this very strong association between conquering this new niche of eating figs and producing many more species. So that was our key finding back then. And that led us to ask the question, well, how are they finding the figs? How are bats, how do you find fruit? How do you find fruit that's ripe at night? And so today, oops, I went in the wrong direction. Okay. So today I'm going to be talking about 3 senses, 2 different kinds of olfaction, as well as vision, right? So the sense of vision, all bats actually have a sense of vision that is, it differs from species to species, and it is definitely a common theme here. And I'm going to be answering 3 very distinctive questions, trying to understand this, how genomic information can illuminate the process of evolution. So for the vision, I'm going to ask the question, does feeding ecology shape evolution? Does this conquering of these new niches, for example, with the figs, does it shape the evolution of the genomic evolution for these bats? And then for one of the chemosensory systems, or one of the olfactory systems, is it possible that this chemosensory system has been gained? And for the other olfactory system, the one that is shared among bats and ourselves, is diversity begetting more diversity? Does diversity in the diet lead to diversity in the genomic variation that we find? So I'm going to start with, does feeding ecology shape the evolution of these genes that are involved in vision? So one of the key things that has happened since Darwin is that, of course, in the origin of species and all his work thereafter, we understand that Charles Darwin did not have a working theory of inheritance. The theory of inheritance that he himself had inherited from the past was not workable. And so he came up with a couple of different alternatives. In different editions of The Origin of Species, you find different versions of how traits are inherited. He was trying to grasp how inheritance could be coupled with. It took another, despite the fact that Mendel and Darwin were contemporaries, and that apparently Mendel mailed his work to Charles Darwin, who was a very notable person already, before publication. Darwin never read Mendel's work and thus missed the theory of inheritance that had to be rediscovered some 50 years after. And then some 50 years after that, so this puts us in the middle of the 20th century, we have this molecular biology taking over genetics in such a way that we actually have this understanding of DNA as the molecule that's encoding the genetic material. And from there, we transcribe into RNA, this messenger RNA that then is translated into proteins. And the proteins end up being, so we have the process of replication, transcription, translation, and the directionality with which this happens. So if we actually want to map this out back to the ecology and back to the interaction, we would have to posit that the sensory information is actually kind of going in the opposite direction, because the sensory information is only interacting with the phenotype. And the closest to the phenotype that we can get to within the central dogma of molecular biology is actually the proteins, right? At the same time, in the process of replication, that's where we're thinking that adaptation is happening, that natural selection is operating on these variants that are being produced through imperfect replication. And that's where the process of evolutionary adaptation is happening. So we actually have these levels at which the ecological interaction is happening, being distinctive from the level at which the adaptation process is posited to happen. And that will come back in a moment. So to begin, the beginning of all these kind of studies, actually field expeditions that we've undertaken throughout the Americas. This picture is from Costa Rica. And in this field expedition, we do our best to preserve tissues in a way that it actually preserves the middle of this, the RNA. We want to preserve the RNA because the RNA, although extremely fragile, it actually gives us information about the protein coding genes that are present in the cells that we are taking from that tissue at that moment. So it gives us a reduced representation of the overall genome, but it still gives us thousands of genes that can be compared across the different species that we're targeting. Our starting point was a rather obvious hypothesis, and this is Kalina Davis, the postdoctoral researcher who spearheaded this paper. So if we have this great diversity of diets that we just talked about a moment ago, great diversity of skull shapes, we can map it onto this phylogeny at the bottom, right, where we have the different species being depicted in the phylogeny. And we see that a couple of times independently, the species, the lineage, the species, and then the descendant species that together make up a lineage have switched their diet from an ancestral diet to feeding off nectar. This has happened a couple of times independently. And we also see that there is this place where it seems that all the bats, all the descendant bat species switched from a diet that was mostly insects to a diet that was mostly plant products. And then there's this other specialization in figs, right? So the obvious hypothesis is that this genomic adaptation that we're looking for in those genes that we're sampling is actually related to the feeding ecology, right? So that every time that there is this switch over to a new diet, there's going to be a burst of adaptation. There's going to be a change. There's going to be evolution such that these genes that are associated with vision are actually going to enable this descendants to actually perform better out in the real world. So that was the hypothesis that we went in. So you can imagine, but when the data came in, and this is data that encompasses thousands of genes across more than 40 species with different diets, right? I'm gonna walk through this plot because this kind of plot is like a very complex, it's the kind of plot that we use to represent data that otherwise would be a very complex Venn diagram, right? So if you look at the bar chart at the top, those are the numbers of positively selected genes. Now, in the language that Darwin wrote in The Origin of Species, he wouldn't have said positively selected genes because they didn't even know that there were genes. Instead, he said there was preservation of favorable variation. So variants that are thought to be favorable, and we use some statistical methods to try to infer which ones they are. And on the plot at the top, right, we have the numbers that correspond, how many we found for that particular group. So we have 429 correspond to the insect at the bottom, to the neotropical noctilionoid ancestor. So the ancestor of all of these bats, that includes the fishing bat, it includes the vampire bat, it includes the fig-eating bats. So we have 429 genes that out of more than 15,000 show the signal of preservation of favorable variation. When we go to the next bar, that's the yellow bar. In the next bar, that corresponds to a nectar, a switch into the nectarivory, and we find 208 genes. But now, we can imagine, since we have two different origins of nectarivory in the same group, we might ask ourselves, is it possible that the same genes get selected for, or that experience positive selection in the two groups? And the answer to that is the box that's in yellow. The box in yellow, we find that only 4 genes, out of all of these thousands and thousands of genes, only 4 are positively selected in both group of nectar feeders. So that illustrates that when you see a line in between the 2 points, that line corresponds to genes that are selected in common, okay? And because genes can be positively selected across a single node or across more than one node, then at the bottom, in the bottom part of the graph, we actually have the sum of the single node plus any other group, any other part that is listed. So bottom line is, most of the positively selected genes do not correspond to the switch to a new diet. They actually, the preservation of favorable variations is instead happening in the ancestor of these bats. So this leads me to have to like readjust. We came in thinking that the switches to the new diet, for example, if this were right, then the green bars should be super tall, because switching to this diet of plants probably demanded from these bats, ancestral bats, a form of vision that they didn't have at the time. And that is not what we find at all. If you look at those bars, they're actually quite low. So this forced us to think in a different direction. And it takes us into this batty aside. So most bat species that we've seen, this is from a GIF, are aerial insectivores. And this is what that looks like. Aerial insectivory, what that looks like is there are insects in the air and they trap them. In the case of this bat that we're seeing, the bat is actually scooping up the insect with the membrane that it has between its legs, right? But there are other ways to catch prey. And the noctilionoid bats, which are the bats that we are focused, the focal group, are not, or most of them are not aerial insectivore. There are some that are, but most of them are actually gleaners. That means that they pick up off the surface. They don't pick up off the air. They actually pick up the surface of leaves, or in the case of the fishing bat, it's picking up fish from the surface of the water. So it's using echolocation to map the surface of the water, but then it's gleaning. And this is called gleaning gleaning. So in the case of insectivores, it's gleaning insectivory. In the case of the fishing bat, of course, it's piscivory that is gleaning. So what's interesting here, what's important to understand is that these bats made a change, not in their diet, but in the actual way that they are hunting. Instead of going in the air, like the majority of bats, they've switched to going off surfaces, like a relative minority of bat species. So we'll get to that in a moment, because when we look at the breakdown of what these genes are doing, so these were genes that were isolated from the RNA, from the retinas of species with different specializations, right? We find that the ancestral bat actually has the greater number of adaptive, of positively selected genes, of genes that seem to be evolving through adaptation. And these genes are involved in mouse model, or they have investigated in human, in the homeostasis of the retina, or responses to salt stress. Of course, the balance of water, maintaining the correct pressure, blood pressure inside the eye is super important, as many of us experience in our own lives, right? And so we see that there's positive selection on a whole host of genes. In the other bats that are actually switching their diet, we do not find as much of a signal. We find a lot fewer genes. And then it bursts up again in the switch to figs, where we have even more. We actually have more genes that are associated with this salt stress and hyperosmotic response. So even though we started thinking that the changes in vision genes were going to be associated with the changes in diet, instead, what we found out is that they're associated with changes in the foraging strategy. The big change, the giant burst of adaptation that we identified these bats is happening in the neotropical noctilionoid ancestor, which is inferred to be primarily switching from this aerial insectivory to gleaning insectivory. To wrap up this section about the positive selection in eye-related genes, we thought that adaptation was following the feeding ecology, but instead adaptation is following the foraging ecology. It's following the style in which they are catching their food. These molecular adaptations, which again, Darwin gave us a language about variation, but not about the genetics of the variation, trace back to the evolution of gleaning rather than to specific diets. And that's really fascinating, because this change in foraging is telling us that that ancestral bat was probably not a specialist at all. It was a generalized bat that was omnivorous. And a few years back, we actually published a paper in which this fossil, the fossil is just a couple of teeth, and this is the scanning electron microscope image of one of those teeth. And based on that teeth, on the morphology of that tooth, we were able to infer what the skull of this bat was like. And in obtaining the skull, we were able to infer, based on observations from bats that are alive today, that 12 million years ago, there was an ancient bat that is shown here that was also omnivorous. So it was a bat that wasn't specialized. It was a bat that could eat insects, but it could also drink nectar from flowers. So it seems like this group of bats, this radiation of bats, for a long time has maintained this ability to forage in different ways that enabled it to find new diets. Okay, so that wraps up that first question. Now we're switching gears away from the sense of vision, which is so familiar to us, to the sense of chemosensation. So a lot of us may be familiar with the fact that of all the senses, the sense of smell is the least understood of all. The sense of, the sense of vision, even to a certain extent, the sense of hearing has received a lot of attention, but the sense of olfaction in humans has been studied only so recently that the publications associated with understanding the genetics of it didn't come until the 1990s. And actually, Linda Buck, who published these first papers, she got her Nobel Prize like in 2004 because of the understanding the genomics of that. So that's how mysterious it is that it's only in the last 15 years or so that or in the last 20 something years or so, that we have a handle on the genomics of that. Whereas the other senses we understood a lot better. This is a cross section of a bat, and we have two different senses of smell in the bat. One of them in pink is the regular sense of olfaction, which is the same that we have. We have only one sense of smell, and it's that olfaction. But there's another one that's shown here in yellow that's known as the vomeronasal organ. Sometimes you find it in books as Jacobson's organs. So we humans, just like all our closest relatives and most primates, have it in some atrophied form. It doesn't work. We have vestiges of the vomeronasal organ, but we don't have a functional vomeronasal organ at all. And my former student, Laurel Yo, has been leading this research for a while now, and she's now doing her postdoc and conducting independent research of her own. But what I'm gonna show is a lot of her work. All right, so we've got these two senses of smell. The sense of smell that we have all experienced, and another sense of smell that we haven't experienced. But I can guarantee you that you've seen that other sense of smell in action. Raise your hand if you've ever seen a picture like this. Have you ever seen this? Yes, I see a lot of raised hands. Yes, what is going on in this picture? Yeah, I see the raised hands, it's awesome. Okay, what is happening? Did you ever wonder, what are they doing? I'm seeing a lot of nods too. Yes, what are dogs doing? Aren't dogs supposed to have this awesome sense of smell? And if they have such an awesome, aren't they supposed to kind of smell something that they can hunt at a great distance? Why do they have to get up close and personal with each other like this? Why do they have to do that? Well, the answer is that they're not using the regular sense of smell, they're using the other sense of smell. They're using the vomeronasal system. And in order for the vomeronasal system, unlike the olfaction that we use that is related to, or it's sensing volatile compounds that are floating in the air, the vomeronasal system has to do with molecules that are a lot less, they're not floating in the air, they're not volatile, right? So that's why your dog is actually sniffing the wall, right? When there's a little bit of pee, they're kind of sniffing the wall right up close because they are trying to smell with their vomeronasal, they're trying to smell these proteins that are present in the urine. And those proteins are giving the dogs and cats and rats and mice, they're giving them signals about the hierarchy, the social hierarchy, the sexual condition of each other, right? So instead of floating around like a volatile, that's what they're using the vomeronasal. So here's a bunch of other mammals that use their vomeronasal. Horses, mice have been studied really well. And of course, dogs, cats, they all have vomeronasals. So this is a sense that's absolutely fascinating to us because we humans don't have it at all. We do not have the ability to use our vomeronasal because it's atrophied. So in most bats, at the top, in the top row, most bats have an atrophied vomeronasal as well. What does it mean for the vomeronasal organ to be atrophied? What it means is that the area, the open area in which the membrane can take some of these molecules from the surface of the nose and travel and go in and attach to the receptors and send a signal has closed up and it's filled up with tissue. There's no way. There's actually no way to get any kind of signal there. But at the bottom in F, E, and G, we actually have some bat species that do have a vomeronasal, that do have a functioning vomeronasal. So this had been characterized across about a couple of dozen species, and there was this mix. Some of them have a functioning vomeronasal and some of them do not. But this is not enough. Why is this not enough? Why isn't it enough to look at like 20 species? Well, this is a picture of my graduate advisor, Dr. Nancy Simmons from the American Museum of Natural History. She got so tired, she tallies up how many species of bats are in the world at any one point. Like she's in charge of that. She got so tired of answering the question that now what she does is she prints a t-shirt, or actually she prints several t-shirts. She wears the t-shirt at the meeting and then auctions it for charity, auctions the t-shirt for charity. So at the time that this picture was taken in 2019, the tally of species of bats was 1,411 different species of bats, right? So the diversity of bats is so vast that having just 20 species is not enough. We need to study more. This is a very brief biochemical pathway of the vomeronasal system. So vomer olfaction is a form of olfaction. There's no, there's not gonna be a test at the end. But what's important is that these pheromones, which are the molecules, right? Those heavy molecules that are present in the pee, right? They attach to a G protein coupled receptor. And from there, there's a cascade of signaling that goes to the transient receptor potential cation channel subfamily C member 2, which is it's better known as TRPC2, okay? So TRPC2 is this key ion channel that enables the signal from the pheromone to get transmitted upstream and into the accessory olfactory bulb, right? So in 2010, Hua Bin Zhao, who is now a collaborator with our lab, in his lab, they had examined what was going on with TRPC2. What does a TRPC2 gene look across a group of bats? And what he had discovered with this little phi in red, this little zero with the cross, is that it was pseudogenized, meaning the gene was broken in most of the species that they examined. But there were a couple of species, literally 3 species, that they had examined that seemed to have a functioning TRPC2. So that cation channel was actually functional. So my former student Laurel decided to expand the sampling because we needed to encompass the entire diversity and particularly try to understand those groups of bats that keep TRPC2. How widespread is this? How common is this? And so we are in the opposite situation than we were before. Instead of looking for the adaptation, we're looking for the rejection of injurious variation. So as you can see, like this is the vomeronasal vomer olfaction is involved in recognizing each other and recognizing the state, the reproductive state of each other. In mice, when you actually knock out TRPC2, you change their fitness. You actually kind of knock out their fitness too, because the males and females do not know how to copulate, how to behave anymore if you knock this out. So in bats, not having double braiding or breaking of TRPC2, the breaking of TRPC2 could be thought of as an injurious variation, but clearly it's not because it's happened so many times. As a result of these analyses, we discovered that it wasn't just a couple of times that it had been lost. Looking at the different ways in which TRPC2 was broken across different groups of bats, there were 13 different losses across our entire sample. But there were 2 families where it was mostly preserved. One is the Phylostomids, which is the American leaf-nosed bats, and another one is Miniopterus, which is a different species from Eurasia and a different group of species from Eurasia and Africa. We wanted to find out whether simulations could help us understand if there could have been a gain, because when you look at a map like this, you could say, well, what happened instead was that it was gained a couple of times. It would be, if we just used parsimony to infer what had happened along the evolutionary tree, that's what we would infer. So we used instead molecular evolution, and we simulated the laws, and what we're looking for is for the dotted lines to actually line up with the simulations, right? So if we simulate laws, the dotted lines and the simulation map out really well. If we simulate gain, conversely, the dotted line for the ones that maintain the function is completely outside of the simulation. From this, we conclude that the inference or the statistical evidence is telling us that loss is the more likely mechanism that is happening here. In other words, the ancestor of the bats had a functioning TRPC2 and a functioning vomeronasal. It could use its vomeronasal probably for the same things that other mammals use it. But then 13 different times it's been lost. It is somehow redundant. And there are 2 large groups that, for reasons that we still do not understand, but are looking for, keep it. So we also discovered that the evolution, the rate at which TRPC2 is evolving predicts the form, meaning that the accessory olfactory bulb, which is the region of the brain that receives the messages that TRPC2 is sending, we could predict the branch length, which is equivalent to how much change TRPC2 has experienced. The longer the branch length, the more likely to have missed an accessory, to have completely lost the accessory olfactory bulb. And the shorter the branch length, which means that there's a lot of negative selection, right? The evolution is actually making sure that things stay the same. The shorter the branch length, the more likely for the accessory olfactory bulb to be present. So far I've talked about TRPC2, about vomer olfaction as if TRPC2 was the whole story. But TRPC2 is not the whole thing. As I mentioned earlier, right, there are these receptors. The interaction that's happening between the environment, the pheromone, is happening first with receptors that are the ones. So and these receptors are called vomeronasal receptors. So in species that have been studied, like mice and lemurs, the vomeronasal receptors help those species recognize each other. If we had a situation similar in bats, then we actually should see a lot of gene duplications, because that is what we see in mice and lemurs. Mice and lemurs, we see, for example, that two species of mice will differ through a lot of duplications. These genes don't evolve, this vomeronasal receptors don't evolve just by changing a little bit in the sequence. They evolve by actually duplicating completely in the genome. So in bats that have a function of TRPC2, we find that the vomeronasal receptors are actually expressing. So let's look at these phylogenies. In red, everything that's in red are genes that have been found in the genome, but they are broken. They are pseudogenized, right? And everything that is in black are genes that we actually found from the RNA of the vomeronasal organ of these bats, and they are not broken, they are functional. So they could actually bind to some of those pheromones. They could actually work, right? These genes have been maintained, even though all of these in red are more closely related, they are still recognizable as being related to genes that are present, for example, in a dog or in a horse. They've been maintained through millions of years of evolution, right? But they've been maintained not through positive selection and adaptation, but through negative selection, the rejection of injurious variation. In other words, for some reason, in these species that are in black, these bat species that are in black, it is injurious for these genes, for these vomeronasal receptors to break down. And so they maintain them. But that is not the case for all species. For a whole bunch of species, the vomeronasal receptors break down and they are alive and well. They just have an atrophy. They're just like us. We have a bunch of broken down pseudogenized vomeronasal receptors too. And so do these bats. Somewhere along the way, this was there was no negative selection associated with this, which is fascinating in its own right. So instead of this expansion that we had predicted, we just find that they're keeping them. They're using this rejection of injurious variations to maintain their vomeronasal receptors. So to summarize, the vomeronasal system in bats is highly conserved in two groups, two families of bats, the Phylostomidae, which are here in the Americas, and Miniopteridae, which is in Eurasia and Africa. Instead of expanding, the receptors are conserved. There's very few of them, and they just maintain their sequence the same. Therefore, it is unlikely that they are involved in species-specific recognition. It's a lot more likely that they are keeping some kind of ancestral function that is very important because it's been maintained for many millions of years, but that we do not know anything about yet. So there's another, another 3 dissertations to be done to try to understand what's going on. So I'm going to get to this is going to be relatively short. The last question that's about, does diversity beget diversity? And as I was outlining before, the perception and chemosensation starts with receptors. And this is a model of an olfactory receptor. So this is a model of olfactory receptor just like we would have in our own nose. These mammalian olfactory receptors get embedded in a biological membrane, right? And these receptors then bind to these volatile molecules, right? They actually, after binding, the receptor activates a switch that starts the mental representation of odor that gets eventually sent to our olfactory bulb that we have. And that is how, and then from the olfactory bulb, that's how we come up, that's how we know that somebody is making popcorn, right? And that's how we know that we need to clean the oven because something's burning inside of it, right? It's through that signaling that starts with the receptors. So the receptors reflect the diversity of odorants. And this has been studied the best of all among mammals in mice. So this is a representation. On the right, we have a representation of the genome of the mouse. Olfactory receptors are the single greatest fraction of protein coding genes in the genome of mammals, right? Something like between 3 and 5% of the protein coding fraction of your genome is devoted to olfactory receptors. And so is the case, right? So there are olfactory receptors, 1,400 olfactory receptors in the mouse. And you can see that they are spread across multiple chromosomes, right? And a lot of them are packed tight next to each other. And this is important because it takes us all the way from the physical arrangement of the DNA to the mistakes that get made by duplication and by occasionally leaving one off. Okay, so that's the representation. Then on the left, what we have is sort of a cartoon of how this would work, right? You have a source, like for example, another mouse or a cat or a fish or a burger, and that source is putting out volatile molecules, right? There's volatile floating in the air, and those molecules are specific to receptors. Some receptors can bind to some of these odorants, but won't bind to others, right? So what we have is that the receptors, the diversity of receptors reflects to a certain degree the diversity of the odorants, right? That is why, for example, there is this vision that for us humans, you know, mice have 1,400 of these olfactory receptors. We have 300, right? So we have just like the pauperate repertoire of olfactory receptors relative to a mouse. But the trouble is that this gene duplication is actually hard to study. The data collection is hard to do because it requires, since they are packed close together and they have all these regions that are relatively similar to one another, then it requires a very high quality genome in which we have the arrangements of them. And this turns out to be rather expensive. We can do it, we can bring the price down to like maybe $16,000, but imagine if you want to do 10 species at $16,000, that's a very big budget very quickly. So the analyses are also hard. The models within species, how it's supposed to evolve and between species are still not fully settled. They're only in progress. So we wanted to scale up and actually go across the diversity of bats, but we didn't have $16,000 or $30,000 for every species that we were interested in. So we came up with a different approach, and this approach is an affordable solution that brings the price down from all of those thousands of dollars to more like $200. And it's called sequence capture, and you can see it at the top. So in this comparison, we've got the sequence capture at the top, the transcriptome in the middle, and a transcriptome is just looking at the RNA from the tissue. We have some Sanger, which is using primers to try to get them, and you can see that the Sanger is actually really, really not very good. And then you have the genome, the one that costs the most. And so we found that we could get like something like 90% of all the markers. We published that last year. About 90 to 96% of all the olfactory receptors for about $200 a pop by using this method that we developed. So we went ahead and we could now turn around and use it in ecologically diverse. We weren't stuck anymore just looking at the vampire bat that's shown here. This Modus rotundus is the vampire bat. We could branch out and do it with other species. So we went ahead and we did it with this gradient of 3 species that they live in the same place, but they actually have very different diets. Carolia castanea at the top, it's mostly pepper species, pepper plants. Carolia sowelli, it's kind of half and half of other, and Carolia perspicillata is the one that eats the least of the pepper plants, and it's the largest of the 3 species. And then on panel B, we have the olfactory receptor subfamilies that have been sampled for them. And from that, we actually measure the diversity. Is the diversity of what they eat proportional to the diversity of their olfactory receptors? So we see that here. There's greater diversity for the generalist. Carolia perspicillata, which is the yellow one, has higher diversity than Carolia castanea when we measure the diversity.
Take the game back whenever you're.
And, sorry, oh, okay. And instead, in the specialist, what we see is that there is a pattern of more of the same. There's a multiplication of olfactory receptors that are very similar to one another, but they do not add to the diversity because they are not different, right? They're instead like, they're just kind of probably doing the same thing. And probably they have to do with the pepper specialization. So to close, we discovered that molecular diversity is related to the dietary diversity of these bats. This is a Carolia bat with the pepper. The pepper plant is the fruit that you're seeing here, and it's happily eating. So what we have is the preservation. It's perhaps an example of the preservation of favorable variations in that having this molecular diversity in the genome enables a dietary diversity, enables the perception of volatile compounds from a range of different plant species. But looking for molecular adaptation of the traditional kind that we've seen some examples before is actually hard. It's complicated because that is overlain on top of a process of duplication and loss where the genes are actually often duplicated. So you cannot match one for one gene in this species and then the other gene in another species, and to say for sure that this is the one that descended from the common ancestor, because in any one of those species there could have been a duplication process. So that complicates things somewhat, but we do have this pattern of diversity begetting diversity. So to wrap up, we had these three questions: does feeding ecology shape evolution? Can we gain a chemosense? And does diversity beget diversity? Well, we find out that yes, the ecology is shaping the evolution, but it's the foraging ecology and not the dietary transitions that are shaping the evolution of vision genes in bats. We also find that in terms of gaining a chemosense, no, you cannot gain a chemosense. Instead, you have many losses, but we open up new questions as to why do the bat lineages that keep it, why do they keep it? We do not have an answer for that yet. And finally, diversity does beget diversity, but is it adaptive? Is this diversity adaptive? It's a work in progress. There's a lot more work to do. Thank you. And thank you to my lab too, shown here in a picture from, I think I'm gonna say early 2020.
Are there any questions for Dr. Davalos? I have a quick question while people kind of get to asking them. And one is, you had two different groups that retained the merocynasal organ. Are those just completely different groups ecologically, or are there any similarities that you're trying to follow up on?
Oh, that's a great question. It's a great question. Superficially, there's no similarities. So I'm gonna go back. I'm gonna go back to that slide and talk about these two groups. So this, yes, okay. So this, can I make my cursor? How can I make my cursor visible? All right. All of the blue dots that are on the right, all of that group is the noctilionoids. So there are these bats in the Americas that include the fishing bat, the vampire bat, the frog-eating bat, the fig-eating bats. All of these bats maintain it, and there's a couple of losses that included one that's associated with the Caribbean colonization. So we have this giant group with very diverse feeding ecology and gleaning. Now, the one in the other side, not the one, most musculus also retains it, but most musculus is not a bat. So the other one group of bats that retains it is the family Miniopteridae, and they are just regular insectivorous bats. They don't have that diversity. They do seem to have a lot of diversity in reproduction, So they do seem to have mating systems that are diverse and reproductive ecologies that are diverse. but no one knows what they're using it for. And no, they superficially, at least just thinking about dietary ecology, they don't seem to have that in common. The same is true for foraging. Their foraging ecology is also not similar.
Thanks. Other questions?
He's already his hand raised.
Yeah. Hello. Nice talk. I mean, it was really interesting. I wonder if your analysis are now moving towards like secondary metabolites, since like everything is related to smell. And I'm thinking like, since phylostomida is at least currently mostly like frugivorous, right? Wouldn't it be like towards on that kind of analysis? I'm just wondering kind of what is your analysis towards to now?
Yeah, so this project that we have here, I actually kind of forgot to mention some of the collaborators in this project. So this is out as preprint. It hasn't been peer reviewed yet, but it's available in bioRxiv. And in this project, we were working with Charlene Santana's lab, and her lab took the volatile organic compounds from the piper plants that are seen here, from different piper plants, to distinguish ripe piper plants from not ripe piper plants, and piper plants that they prefer from non-ripe ones. And then we made artificial, well, we is really Charlene Santana's lab, made a fake fruit with the organic compounds that we think, based on those analyses, is the one that tells the bats that I'm ready to be eaten, I'm ripe, right? Or the signal for that. And we need that to get accepted somewhere so that everyone can read it. But it's definitely the direction where we're going. It's definitely where we want to go, is to have this matching, because the olfactory receptors, you know, even in humans, even in mice, most of the olfactory receptors haven't been characterized. We know they're there, and we may know what their structure looks like, but we don't actually know what they bind to. So there's a wide open frontier of hundreds of genes whose functionality remains to be assessed, right? We know that they are an olfactory receptor, but we don't know like which volatiles is it important for. Yeah.
Thank you.
Any other questions? I did have a couple more, and one of them was with the sequence capture technique you're using. There are these long read sequencing methods now. Is that something your lab is exploring too, or is that just not going to add any more information for the type of research you're doing?
For the sequence capture that I'm presenting here, we only use short read, but we only use short read. Not that, I mean, of course, we actually at first tried to use long read with primers, but that wasn't very good at all. Actually, both the needed coverage and the primer bias was too strong to be actually able to get anything that was very diverse out of it. So we went with the short read sequence capture. There is another work that I didn't present that one of my students was co-advised with Pat Wright, who works with lemurs, right? And so for that, we were interested to see if we could capture the gene, but we can also get the promoters. And so we used long read. So we used also sequence capture, designed for sequence capture, but long read, so that we could try to get extra bits of the genome capture.
Okay, great. And
yeah.
The other question I had was with the foraging. I thought that was pretty interesting. The difference between feeding and foraging, and a lot of studies that we see published with fish and communities are just based on diet contents, and it made me think about like the importance of the actual way in which organisms are capturing. So was there an did you have a chance to look at the morphology of the bats as well, and look at, is there a strong association between the morphological variation and the foraging strategies?
There is, and that is a completely different paper because this was called a Genomic Journey. Let me see. Are people still seeing my screen? Am I still projecting? Okay, so
Yes.
you get to see all of this. Let me see if I can find that paper. So we actually also did that work, and this is work out of Betsy Dumont's lab, right? But we were collaborators in this. Is it this one? Yeah. So let's see. So we were looking at the diversification. And so you once more, you've got this tree where this is the fig eatings and the carnivores, etc. And you can see those shapes, right? You can already see like the shapes of the skulls are different. But we also had like this three-dimensional, I know that Windsor, this is something that you've worked on. So we also have the CT scans for the whole skull and looking at the morphological space that they're occupying. And we see a bit of that morphological space. Let me try to make this bigger. More of that morphological space here. And what we find is that the phyllostomins, which have that huge dietary diversity, they are across this axis. So these are the mormopids, which are aerial insectivores. They're descended from their sister taxa, but they go in a completely different direction that's perpendicular to the direction that the phyllostomins go, in which they're going. So, and these are very distinctive from their relatives that aren't noctilionoids. So there is the foraging ecology is related to a new arrangement of the skull that seems to make it more flexible. And I just encourage you to look at this paper. Let's go look at this paper. Diversification under high integration in a hyperdiverse mammal clade. If you want to have a look at that, yeah.
Thank you. Any last questions? Last chance.
Oops.
Okay, well, don't go yet because I did want to say just a couple of words. One is I hope there were, I hope the graduate students appreciated and the undergraduates that came appreciated the span of what you talked about with, talking about aspects of their ecology and then linking that to a phylogeny, which we try to emphasize in our classes, and then looking at the power of genomic data to try to answer pretty important questions about speciation. I think it's a beautiful representation of that. So thanks for doing that.
Oh, thank you, Windsor. I don't know that I would have looked at it that way.
Thinking more, I just finished my part of teaching one of our biology classes where we covered some of these things. But and so thanks for that. And also just can I share my screen, Carolyn? Okay, let me share my screen here. Oh, I'm sorry. So just to present you with a virtual Darwin Day certificate for being our speaker and taking the time to do this and giving such a wonderful talk. Are you okay with Amazon, Liliana? How do you?
Amazon.
Amazon.com.
Sure. I mean, how
shoot,
could I not? I mean, it's a pandemic. Okay. Like so everything is being delivered.
Sending you a gift certificate as well.
Really? Oh, wow. Yay. I'm very happy.
And thank you very much. And yeah, with that, I will let everyone disperse to their 3 o'clock meetings.
Okay, well, stay tuned. We have a whole bunch of things to publish on this topic, right? Some stuff is out, but there's a lot more to go. Okay. Thank you, Windsor, and thank you, everybody.